pancreas. That, of course, the sodium is removed and you're left with bicarbonate, and that is involved in this carbonic acid bicarbonate buffer system. We mentioned the buffer system, when we were talking about things in lab on last Wednesday. The buffer system is really important because our pH range is 7.45 to 7.35 to 7.45, really narrow range that we can tolerate. After that, when we go too high and we go too low, the same damage can occur. We don't want to be metabolically alkaline, we don't want to be respiratory alkaline, we want everything nicely balanced in that very narrow little range where we're kind of neutral. 7, right, on the pH scale. What else happens in this region of the pancreas? So the pancreas produces these proteases. We can't make trypsin in the pancreatic cells because why?
Digest itself. It would digest itself?
Yeah, it would like burn up the cells that are making it. So we can't make trypsin, we make trypsinogen, which is a precursor, a prozyme. Do you remember the other prozyme in the stomach?
Pepsinogen.
Pepsinogen. What cells produce pepsinogen?
Chief cells?
The chief cells. And what does that pepsinogen do?
Protein digestion.
Pepsin. It becomes pepsin.
It becomes pepsin
Pepsin.
in the presence of
Hydrochloric acid.
hydrochloric acid.
Or
Exactly.
Or other pepsin.
Or other pepsin. Good, because that was the next place I was going to go. So when you have pepsinogen converted to pepsin, and then that pepsin can convert more pepsinogen, what's that process called? Autocatalysts, autocatalytic activity. Well, the same thing can happen with trypsinogen. You make trypsin, and then trypsin can convert other proteases. But before we get to trypsin converting these guys, we have to remember that trypsinogen is turned into pepsin in the presence of enterokinase, which is the same thing as enteropeptidase. I know. 2 names for the same thing. Enteropeptidase is produced where? Endocrine cells. Where are they located? Small intestine in the crypts.
Yes.
Right, the intestinal crypt, you got those little divots and those cells down in there, and they're making the enteropeptidase. That enteropeptidase then can convert trypsinogen to trypsin, and then trypsin can change chymotrypsinogen to chymotrypsin, and procarboxypeptidase to carboxypeptidase, and elastin, proelastin to elastin. So we have this big, wonderful conversion just by changing one enzyme. You can convert all these other proteases that are going to take your complex proteins, your tertiary structure proteins, and convert them into peptide chains, and then you get dipeptides and tripeptides, ultimately get converted to amino acids. So cool, that's what happens in the pancreas. The other accessory organ for the digestive system is the liver, and you're well familiar with this appearance now. You go your right lobe and your left lobe, and on the backside you've got caudate and quadrate lobes. So 4 lobes. You have a ligament running down the center separating your 2 big lobes called the falciform ligament, which becomes known as the round ligament once it leaves. And this, by the way, is the remnant umbilical cord. Do you remember looking at the circulation of the fetus and seeing the ductus ligamentosus, right? That's this. That was that. Yeah, so the blood getting carried up to the heart via a passageway through the liver. So we have these big lobes. The big lobes are divided into what we call lobules, and each lobule is surrounded by structures at the corners. What are those structures called? The portal triads, exactly. And those portal triads are very important because they are going to supply the functional units of essentially the sinusoid structure is the functional unit of the liver, with the portal triads containing 3 basic structures. What are the 3 structures contained in each portal triad?
Bile duct?
A bile duct or ductule, they're really tiny. So bile duct, ductule. Yeah, what else?
Branch of the portal vein.
Branch of the portal vein, and what else?
Hepatic arterial.
Hepatic arterial. You could say hepatic artery, portal vein, I'm happy. that you got all that. Obviously, it's small, so we could say venule and we could say arterial and still be accurate. So what happens with the liver is that the blood from the intestines is delivered to the liver. What are the three blood vessels? Two major ones, and then the third one that carry blood from respective structures to the liver, to the portal, that merge to form the portal vein. That's what I was trying to say.
Okay.
Yeah, sorry. Yeah. So what are the main veins that are delivering this nutrient-rich blood? Not much oxygen, but a lot of nutrients. One begins with S, and then M. Did I hear superior? Mesenteric, I thought so. Superior mesenteric artery, or vein, excuse me. And then what's beyond the superior mesenteric vein? The inferior mesenteric vein, yep. And the splenic is like another main vein that feed into the portal vein. Then the portal vein delivers that nutrient-rich blood via portal, right, part of the portal ion, to the sinusoids. What are sinusoids?
Capillaries.
Waveforms.
Capillaries. What's special about them?
The large openings.
They have large gaps, and so big things can pass through those gaps. So we have sinusoids, capillaries, that are bound on either side by hepatocytes, and all over the place the hepatocytes are located, and little canaliculi, bile canaliculi. The hepatocytes are busy processing all that nutrients, all the nutrients delivered to this lobule via the portal veins. All those nutrients are extracted from that blood, used to build new proteins, to build so many things and break things down. 200 plus jobs is what the hepatocytes do. Our primary concern is that those hepatocytes produce the bile, which is then delivered to two primary hepatic ducts. You got the right hepatic bile duct, you got the left hepatic bile duct. They merge to form the common hepatic duct. And then we have this other known structure that's part of this whole process called the gallbladder. And if the bile is being produced in excess, it will be stored in the gallbladder. And then when it is called upon, you eat a big fatty meal, burgers, French fries, what else? Chocolate cake for dessert with a lot of frosting, lots of adipose, a lot of fat, a lot of triglycerides. Then that bile is going to help do the processing of that. And by the way, you want to remember that the bile emulsifies the fat, so it reduces the big globs to smaller globs. And those smaller globs are then surrounded by the bile salts, which allow for exposure to lipase. And then the lipase comes in and hydrolyzes the triglycerides, reducing them to fatty acids and glycerol. And the reason we saw a change in the pH in our experiment in lab was because the fatty acids reduced the pH, and hence we saw a bigger change. Bile, lipase, lipids, and the perfect body temperature, 37 degrees. There we should have seen the biggest change in our pH indicator. Not that it can't happen without, it just takes a lot longer. And you want to be at just the rate of everything. So, once that blood percolates through the sinusoids, it ends up in the central vein. These central veins merge to form hepatic veins. The hepatic veins then dump into the inferior vena cava and back up to the heart. Back to the lungs, back to the heart, back to the body. And around and around and around we go. So that's the basics for the liver. And here's some of the things that the liver does for us. We'll talk about that. So we know about the bile production now. And we also know that the liver is capable of storing and releasing glycogen, just talked about that. The liver can make lipids, and it can store vitamins, and it can store minerals, very important for storage. And then we have the ability to convert glycogenesis, is making glycogen, so taking excess sugar and excess fat and converting it into glycogen. Glycogenesis. We can make glycogen, we can turn glycogen, into glucose. We can make glucose from lipids and proteins. This is really kind of important. It's one of the things that you don't necessarily want to do, but if you have excess glucose, you're going to be turning it into lipids or proteins, mostly lipids, and stored in adipose tissue. This is what happens when you eat a big meal at the end of the day and you don't burn that glucose. It's going to get converted into fat and stored as extra energy. The deamination to make glucose, we're going to talk more about that when we get to the metabolism discussion. Lipogenesis, so we can make lipids and we can break lipids down. And this all can happen in the liver. The liver can make up plasma proteins. The liver can detoxify things. It can detoxify drugs, get rid of hormones, get rid of all of that excess stuff, send that off to the circulatory system where the kidneys are going to take care of it and get rid of it entirely. Phagocytosis. So we saw in lab there was this mention of the Kupffer cells, which are the phagocytes that live in and around the liver. We have a great slide that has them, the great slide that has them stained especially is in histologyguide.com. We're not going to ask you to identify those Kupffer cells on a slide in the lab, but you should know that they exist and that their primary job is phagocytosis. And obviously, anywhere around the body where we have blood, we need to be capable of dealing with the potentials for things being delivered to, like bacteria and virus-wise. So, nice close-up view of our portal triad. Here is the portal vein, here's our hepatic artery, there's our bile duct. We have the little canaliculi tucked in between the hepatocytes, so they're processing and producing the bile, and that bile is going to be then delivered to the intestine and also your gallbladder. Yeah, pretty amazing. And I like to remind everybody, your liver is busy, busy, busy right now. Very busy. Building up, breaking down, taking care of stuff you don't even have to think about. We're lucky. And another really good thing about the liver is you can take a chunk of it and it will regenerate if the chunk is small enough. And you can lose a chunk of your liver and still exist, still survive. Yeah?
But it's not like infinite, right? Like, you can't just keep taking a chunk out like every year and just like, expect it to kind of grow.
No, I don't think you can do that. Yeah. Oh, disease. Yeah. Cancer. Although I've never heard of anybody survive cancer of the liver, because it pretty much has metastasized at that point in the liver. I know just one person who died of. I know two people. Two friends died of cancer of the liver. Liver cancer. All right. So the next to talk about on a much more pleasant subject is the gallbladder. The gallbladder and the storage of bile. And so here are our right and left hepatic ducts. There's our common hepatic duct. Here's our gallbladder. The gallbladder also has a head, a body, and a neck. And coming from the neck is the cystic duct. And the cystic duct is going to merge with the common hepatic duct and form the common bile duct. And then the common bile duct delivers its product to this inflated ampulla. So there's our ampulla. And there is the sphincter that's going to prevent all of this stuff from just being delivered, massively into the duodenum. And then we're going to control it with the sphincter of Vater. Here's our major duodenal papillae. So it's just this raised area surrounding where the hepatic duct and the common hepatic duct and the major pancreatic duct merge. Here's our minor duodenal papillae. I guess there are some people that don't have both ducts, just like we have different arrangements for the blood vessels coming off the aortic arch, and we have different arrangements for our kidneys. We have differences in people. What else? Contraction of the gallbladder, of course, is going to send that bile down into the ampulla and deliver it into the small intestine. And that bile is important, obviously, because it enables us to process lipids very efficiently. We can live without our gallbladder, and we can live without that extra bile. But the point is that if you lose your gallbladder, or if you have gallstones and they're impeding the storage of bile, you might want to cut down on the amount of lipids that you're eating, because if you don't, those lipids are just going to pass through the intestinal tract. And we need lipids, obviously, for our cell plasma membranes. We also need lipids to carry those lipid-soluble vitamins, A, D, E, and K. And so if we don't have the lipids to get those vitamins into our system, we don't get the vitamins. And those A, D, E, and K have significant impact. Like D, right? Vitamin D. So bile is great when it's working well and our gallbladders are working well, but bile can also develop into these little concretions, these little nasty little balls, or they're not even all balls, but they are bile salts. They are a combination of cholesterol, and you got some calcium, and the production of these gallstones can impact the ability for your gallbladder to store bile, but also to deliver bile, because we need to have this open in order for the bile to be delivered. So imagine if you have a big stone sitting right in there, then you have a big problem. So you're going to have your gallbladder removed, then you're going to manage your lipid intake. Gallstones. The cool part about bile, which I love, approximately 80% of all the bile that is produced is recycled. So you use the components and you make more bile out of it. Cholesterol, we make a lot of cholesterol. Our body is essential, cholesterol is essential for managing our steroidal hormones. Cholesterol, chole, bile, sterol, steroid. We need our cholesterol, we need lipids, of course, for our cell plasma membranes. And as I was saying earlier, we got to make sure that we can get our vitamins into our bodies. How do we manage our digestive system? Because you are sitting there, you're digesting stuff. I don't know the last thing you ate or how long ago you ate or drank, but obviously you put it into your stomach and you forgot about it. And you have to think about it again. And that's because we have a very comprehensive mechanism for managing the food that we've taken in. So you might remember the different kinds of regulation that we have operating in the body. We have autoregulation, also known as intrinsic regulation. Where is that? What is that?
It's like stomach digestion, stuff or not.
Like happening right there at the site. Right there at the site. I always think of smooth muscle, and when smooth muscle is stretched, it contracts. That to me is the ultimate autoregulation. And then we have extrinsic regulation, and I think it's very clear that we depend on our nervous system. And what kind of nervous system are we depending on?
Enteric nervous system?
The autonomic nervous system. Yeah, the autonomic nervous system. We don't have to think about sympathetic, parasympathetic motor innervation. And when we are big meal, sitting back, watching TV, or whatever, playing whatever you're doing, scrolling on your phone, which is probably mostly what's happening these days, you are dependent on your parasympathetic system for driving your digestive processes. That's where we want to get blood to the intestinal tract so we can take all those nutrients, get them to the liver, get them, working for us. So we have the neural aspects and then the endocrine. What part of the endocrine system are we dependent on for digestive regulation? We just talked about the pancreas is a biggie. And then all the way down the intestine, you have the brush border enzymes that are making those enzymes, doing their job right then and there, which is almost autoregulation, but no, it's actually endocrine because we're making enzymes. And you got those enteroendocrine cells, also part of the process. So autoregulation, where things happen right there at the source. This is describing how histamine is released from these enterochromophilic-like cells. The stomach gets the parietal cells going, the parietal cells to get their job done. What do the parietal cells produce again?
Hydrochloric acid.
Hydrochloric acid, yep. Also, no. And then the stretch, as I was saying, first and foremost. Remember talking about the nervous regulation? We talked about the central nervous system and the peripheral nervous system. And then in the peripheral nervous system, we had the somatic and we had the autonomic. Somatic is voluntary, autonomic involuntary. And we began to discuss in more detail the autonomic system, sympathetic, parasympathetic. And then they also suggested that there might be a third portion of that autonomic system called the enteric nervous system. Remember discussing that? And then we said, well, we're not going to do that because we're just going to stick with autonomic, sympathetic, parasympathetic. But it does seem to lend itself to think about having an enteric nervous system because we have these two layers of fibers, nerve fibers, between our circular and longitudinal layers of our smooth muscle, and also sitting just in the submucosal region. And so we have this really comprehensive mechanism for controlling what is being moved through the system at any given time. And apparently there are a couple of different reflexes that are going on within the digestive system, the enteric nervous system, which is right here, and then also connecting it with the central nervous system, because you know that you have the ability to control defecation. And that comes from up here, cerebral cortex, where you say, it's not a good time, I gotta hang on. But other times, you're ready, you go. So this working with the central nervous system is called a long reflex, where you have the pressure, you got things moving, and then up to the brain, and your brain says, got it, let's wait a while for all of that pressure to be dealt with. There's also short reflexes, and those short reflexes have everything to do with getting this smooth muscle contracting. So you put food in there, you get a big bolus of fluid coming through your small intestine, it stretches that smooth muscle, you get the contraction. That's the kind of short reflex that we're talking about. So long reflexes involve the central nervous system, short reflexes, sort of right there, Johnny, on the spot, to the spinal cord, back out again. And what is it that we're looking at? Sympathetic, parasympathetic, right? And in this case, digestion is all about parasympathetic innervation. Sympathetic, we don't want to have when we poop in our pants while we're running away from something, right? Now you might have the initial empty your rectum, but then you're not producing any more. Everything is going to be tamped down until you're through your crisis. That's the way that works. Okay. Now, we do have some hormones that we got to talk about that are essential for this process that we call digestion. The endocrine cells are producing these hormones, lots of different peptides that we're getting to work on here. Did you know this, that if you see something that's ending with an IN, it's generally going to be a protein hormone? Have you ever heard that before? You might want to tuck that. Have you heard that, Carla? Yeah, tuck that into your little memory bank. So I probably said it before. Yes, it's just a little help aid. So you see this word, you're not quite sure what it is. If it ends in IN, it's probably a protein hormone versus a steroidal hormone. Remember that from 232? You got your hormones that can be diffused right across the plasma membrane, and then you have a whole bunch of hormones that need that second messenger system to get their message to the inside of the cell to tell the cell what to do. Furrowed brow. What?
The first and the second carriers, or
Oh, the G protein.
okay.
I'm thinking of G protein.
Okay.
Yeah, and then adenylyl cyclate.
Okay.
But you just have to remember, second messenger system.
Okay.
Remember, it's the first person comes to the door, has the mail, puts it through the slot in the door, the dog grabs the mail. The male person is the hormone, second messenger system delivering the mail through the slot. That's the G protein. The dog grabs it and down the chain of proteins to tell the cell what to do. That's you pick up the letter from the dog and you open it up and you're told what to do. Report to. Yeah. Okay. So here are some of the hormones that we need to pay attention to. And I looked at this word, cholecystokinin, or CCK, and tried to put it all in my brain. And that's why I bolded things, because if we have cholecystokinin present, it's going to tell the gallbladder to contract. Cholecystokinin. Cholecyst. Where's the cyst part?
Cystic?
Well, a cyst is a balloon-shaped fluid-filled structure. So we have a gallbladder that is actually a cystic structure. Our bladder is a cystic-like structure. Hence, they call it cystitis when you get a bladder infection. So we have chole. This is the cholesterol part. Think fat. Cyst from the gallbladder. Protein hormone. So the gallbladder is stimulated to contract. If the gallbladder is stimulated to contract, and this hepatopancreatic sphincter is allowed to release, we're going to get material from the pancreas and the bile duct into the duodenum, and this all indicates that you have food in the system. And if you have food in the system, then cholecystokinin is going to tell you you don't need to eat anymore. So it's going to suppress your appetite. Once you have food in the system, there's no need to eat more. Inhibition of stomach secretion and motility makes a lot of sense too. Now you've processed everything, you produce the chyme in the stomach. That fluid, that chyme, is going to be squirted into the duodenum in small increments. Does anybody remember the volume that's approximately squirted? 2 to 3 mil. Yep, every 2-3 seconds or minutes, depending on what it is. If it's a fatty-based food meal, probably slower than a watery-based food meal. All right, so inhibition of the stomach, because you've already got food, you got to get that material from the gallbladder and the stomach into the small intestine. That's what these guys do, and then stopping the process of producing any more. We already got food in the system. Secretin, just as its name implies, is all about getting bile into the liver, from the liver, so bile secretion from the liver, and getting the pancreas to secrete its pancreatic enzymes. And if all of this is happening, then we don't need any more stomach secretions. We already processed a meal, so a reduction in stomach secretions. Appetite suppression from cholecystokinin, secretin stops the secretions because we already got secretions going on. Glucose-dependent insulinotropic peptide, oh my God. GIP, we're going with GIP. We know that it has to do with insulin, exactly this, insulin secretion. This GIP has to do with insulin secretion. And vasoactive intestinal peptide, or VIP, to dilate the capillaries in the intestine. What's the value of that? Well, now that you have your food in the intestine, you want to be able to get those nutrients into the circulatory system. So let's dilate those capillaries, make them a little more accessible to get those nutrients into the circulatory system. And all of that blood is venous blood, deoxygenated blood. All of those capillaries are going to merge and form those superior mesenteric vein, inferior mesenteric vein, and all of that nutrients is going to be delivered to the liver by the portal vein. So we want to remember these hormones. Cholecystokinin, secretin, GIP, and VIP, and this is a better way of memorizing it all. That we have gastrin produced in the stomach. Gastrin production by the parietal cells, gastrin production by the parietal cells, stimulating the parietal cells rather to make acid, their hydrochloric acid, and that gastrin is going to actually be stimulated a lot by your cephalic phase of swallowing, the cephalic phase of this whole process. So you think about food, you smell food, your stomach starts gurgling, it's because you release this gastrin from the stomach through the circulatory system back to the stomach to stimulate muscular contraction and the juices, production of juices. And this is why you want to eat, because if you don't eat, then you have all that hydrochloric acid with nothing to do. And you can burn through that mucus, so you can get gastric ulcers if you don't eat enough. So we got the gastrin, then we have the GIP, the glucose-dependent insulinotropic peptide GIP releasing insulin, stimulating the release of insulin, which of course is going to be compounded by the presence of blood in the, yeah.
Okay, so just so I know, I know that I'm right. The cholecystokinin suppresses appetite, and the gastrin stimulates appetite.
Correct.
Okay.
Yep. So, and the gastrin happens well before the cholecystokinin.
Oh, okay. Yeah,
So
that makes
once you
sense.
bring food into the stomach, then
Yeah.
the cholecystokinin says, okay, you've got food to process, we don't need any more food in the stomach. So we're gonna reduce the secretions and,
That makes sense.
yep. So secretin and CCK, right, getting your pancreatic enzymes released, getting your bile released from the gallbladder, and then you got your VIP dilating those intestinal capillaries so you can absorb the materials that we need to absorb. Okay? And then you're going to use all those nutrients for all the tissues. So the liver builds and breaks down and builds and stores, and amazing organ, the liver. While you're sitting there. There we go. Next to talk about is how we move all this stuff through our process, our digestive process. We have these reflexes to consider. The swallowing reflex has 3 different phases. The swallowing reflex starts with the oral phase, where you put food in your mouth. Have you ever put food in your mouth and then not done anything with it? Yeah?
Sometimes I've been like just chewing on it.
You are chewing on it, so you are doing something.
Okay, yeah.
If you put food in your mouth, what do you start to do? You start chewing. Right? What? What'd you say, Omar?
I said open your mouth.
You open your mouth, and then you put food in it. Put food in your mouth, and you start chewing. This is a reflex. It's a voluntary reflex, but I can't think of anybody that I've never put food in my mouth and just let it sit there.
Hard candy, you'll sometimes suck on
But
it.
you're sucking on it.
Sucking on it,
But not, yeah.
What if you don't like the food and you're like, oh.
then you spit it out. Okay, you could do that. But then we don't have it, we don't have to talk about any other reflexes, so.
Now I put food in my mouth and not do
Right,
anything.
so you put this chunk of food in your mouth, you start chewing it. This is your first part of the swallowing reflex. You put the food in your mouth and you start chewing it and chew it, and some people have like this number of times they chew. If you heard, some people are suggesting that you chew 20 times, or I don't even know how many times. Like make sure that you chomp on it plenty of times to break it down as much. Have you heard that? Like how many times have you told
Oh,
to chew?
I'm not sure until, I don't know, I just heard that you're supposed to break it down past the mush parts so
Yeah.
that it digests better and, yeah.
Yeah, Carla?
I watched a video after we were going through the digestive system.
Yeah.
It said something like soft foods is 10 to 15 chews, and then like going from there, medium is like 20, and then hard.
So I was right, I was following 20 or so chews to make sure you process that food in your mouth. And then you send that food to the back of your mouth to a region known as your oropharynx. And by the way, and this kind of always surprises me, that we have the trigeminal and the glossopharyngeal nerves that are being signaled to get involved in this voluntary chewing process. We know the glossopharyngeal is motor to the tongue in part, but also sensory. And also the glossopharyngeal is motor to your pharynx. So we have little muscular structures called the pharyngeal arches. While they are a skeletal muscle, they're still involuntary, and they are part of this process of sending that bolus of food in your mouth back to your oropharynx. And that stimulates the swallowing process So it's stimulating, you have a nuclei in your medulla stimulating the swallowing process. You're sending it back to the back of the tongue via the glossopharyngeal, and you get that bolus back here, and boom, you're going to swallow it. There's no stopping it. And that's known as the pharyngeal phase. And this is the part that sends that bolus down the esophagus, which is then known as your esophageal phase. And literally, the esophagus is a conduit. It is skeletal muscle at the upper reaches, more superior, and then emerges and becomes more smooth muscle when it gets to the stomach. And there's really nothing else that happens with the esophagus except for that peristaltic action. And so we've been back and forth about the esophagus being part of the mechanical digestive process. It's certainly not that chemical digestive process, and I don't want you to sweat about it because I'm never going to ask you. What you need to know is that the esophagus is a conduit. It's going to send that bolus down to the stomach. And it's going to do that utilizing peristalsis. And it can do that even if you're standing on your head, which is kind of crazy, right? And it doesn't take very long for it to get down to the stomach. Other things that happen in this pharyngeal phase, don't want to mess that up, is that when you swallow, obviously that bolus of food, you want to make sure it clears your trachea. It doesn't go down through the glottis, and it doesn't go down through the glottis because you have a little flap called the epiglottis. But it's not that the epiglottis literally comes down like this, but rather your larynx is lifted up as you're swallowing, and it sort of meets the epiglottis, more than halfway. You also have the vestibular folds, which are closing the glottis, so that there's very little chance you're going to inhale that food. Now, if you're talking and you're laughing and you're having a great time and you're eating at the same time, it can happen, right? Who has not inhaled a little saliva at some point in their life? And what happens when that happens, when you inhale saliva? Oh, big time, right? Yeah, big time, big coughing. Okay, so another thing that happens, don't want to forget about that, is the upper esophageal sphincter has to open up, and then the upper esophageal sphincter closes to prevent any of that from backflowing back up into the pharynx. And then we have the gastric reflex. And honestly, I think it's just easy to think of this in three phases. You have your cephalic phase, you have your gastric phase, and you have your intestinal phase. And easily to understand that your cephalic phase is all about thinking about the food. You're so hungry. What happens? Your mouth starts salivating. What happens to dogs when you bring out the dinner? Drool, right? Pavlov's dogs heard the bell, started drooling because they connected the bell with him delivering the food. We're kind of like Pavlov's dogs. You smell the food, you think about the food, you see the food, you start drooling. And gastrin has been released and has set up that stomach motility and the release of those gastric juices. And now you want to get some food into that stomach. Absolutely. Because you don't want these gastric juices working on your stomach walls without food to work on. This is referred to as a long reflex. Some things happening in the stomach are reflexes. It's because of cranial activity, your cortex, your cerebral cortex, gets you thinking about how you're going to get this food processed. A long reflex. Your chief or parietal cells are busy, you're making that histamine, you're making the gastrin, and your stomach is getting going. And so your stomach is ready for the food. Once the food arrives, then you get increased motility, some more mashing of that food. Makes total sense, mixing that food up with the secretions. You get your release of the pepsin that's going to start to break down your complex proteins into polypeptide chains. And those polypeptide chains will be further reduced to amino acids as they hit the proteases from the pancreas. Autoregulation. What's the autoregulation part going on here?
Stretch and smooth.
You stretch the stomach. You add food to it. You stretch the stomach. Smooth muscle wants to contract when you stretch it. Yep. And then your hormonal regulation. Everything is happening through the gastric phase, when the food hits the stomach. Lastly is your intestinal phase. The intestinal phase is the arrival, as this says, of chyme from the stomach into the duodenum. And that food that's coming from the stomach, remember, is acidic. And I don't mean food, I mean fluid. It's very fluidy and very acidic. And so we have then the pancreas and the sodium bicarbonate from the pancreas that helps to reduce that acidity, raise the pH. You have the duodenal glands that are going to raise the pH back to more of a manageable pH, like 7.5 and 8 even, quite a bit more on the basic side. Some other things that happen is that your lower esophageal sphincter has opened and then it closed. Now the stomach is full. And then the pyloric sphincter opens and squirts that chyme through. And then finally, once the duodenum is engaged and you've moved the fluid from the stomach into the duodenum and all of that fluid has been moved, then the pyloric sphincter stops working because we don't need to be squirting any more gastric juices into the duodenum. Now we want to work on the stuff that's coming into the intestine. And there's where our hormones, CCK, cholecystokinin, secretin, and GIP are going to engage. And this is just another mechanism for looking at it so you can read through it. But remember your three phases: cephalic phase, gastric phase, intestinal phase. And pretty simple to appreciate what happens in each of those phases. And then lastly, we have the defecation phase, the defecation reflux. So we produce this waste product called feces as a process of extracting all the nutrients from the foods we've taken in. Hopefully we've eaten plenty of green leafy vegetables so that we have plenty of fiber in the form of cellulose, because we know we don't process cellulose very well. And that cellulose is really, really important because that cellulose adds bulk to the waste material that you're producing in the large intestine. And the bulk of that material is going to stimulate the large intestinal smooth muscle cells to contract. More peristaltic action to move that material down the length of the large intestine. Fiber is very important for creating bulk, but it also scours the walls of your large intestine, so it keeps things clean as well. So, eat your green leafy vegetables, Jack Toland. Do you know somebody that doesn't like greens? I do. You don't like greens?
No. I eat them because I have to, not because I want to.
Because you know, yeah. I love them. But I'm not an enthusiastic cook, so.
What's your favorite?
My favorite greens? Oh gosh, I have so many of them.
Collards.
I like all of the broccoli family. I like cabbage, I love cauliflower, Brussels sprouts, I love them. I like the squashes a lot. Oh, but they're not green leafies. Green leafies, I like spinach. I like it all. Kale, not so wild about kale. But I'll eat it. Yeah. All right, so back to the defecation reflex. All right, so we have a region of the large intestine called the sigmoid colon. Sigmoid colon delivers that waste material to the rectum, and the rectum stores it. And remember this thing about the bulk movement, the mass movement, so 2-3 times a day, you just have this massive movement of material in the large intestine that ultimately puts fecal material in your rectum. The rectum is going to lead to the anus, and the anus is surrounded by an internal anal sphincter and an external anal sphincter. When we have a full rectum, we have pressure on the walls, stretches the smooth muscle. That smooth muscle stretch goes to the spinal cord, and then you have a synapse where you have motor driven, right? Parasympathetic to move this fecal material out of the rectum and out of the anus. But you also have information coming from the cerebral cortex saying, I feel it, but I'm not ready. This is not a good time. And this is what you're teaching children, to detect that feeling of pressure in the rectum. That then tells that child to go and sit on the toilet and empty the rectum. Interestingly enough, this smooth muscle, internal anal sphincter and the external anal sphincter work opposite each other, not opposite, but differently. So the internal anal sphincter is smooth muscle, external anal sphincter is skeletal muscle. This one will be released ultimately because of that pressure, but the external anal sphincter is under voluntary control until such time you have so much feces in your rectum or it's really watery and you have diarrhea.
So I was about to ask about.
Yes, then it's going to leak through the sphincter. But what you learn to do when you learn to manage your bowels is you learn how to manage this external sphincter. You learn how to relax it because it is constantly in contraction. You know that feeling because every single one of you in here defecates at least once a day. I'm hoping. Because my mom said, you gotta poop once a day. She didn't use the word poop because she was a children's nurse. You've gotta go number 2. And that, gang, is the end of the digestive system. And they've got tables, all the things that you need to know about each of those processes that we talked about. Do you have any questions? Yes, Carla?
You said that external sphincter is,
Voluntary control. Yep. Skeletal muscle. Voluntary control. Unless you have a full rectum and you are in a crisis mode, you suddenly need to defecate. But you're not going to be making any more. You're just going to save that and slow everything down because your parasympathetic system is not engaged right now. Your sympathetic system gets engaged. Now, blood going in the intestines, everything slows down because all your blood's going to your skeletal muscles. So you can climb the tree and get away from the predator. All right, so we're not going to stop here, guys. We're going to break into our discussion about metabolism.